It's a pleasure to have with us today Professor Kamil Kranc. He's a full professor of hematology and deputy lead at the Center for Hematology Oncology at the Barts Cancer Institute in London. The aim of his laboratory is to understand the biology of leukemia stem cells, and most important also to identify therapeutic targets to eradicate these malignant cells. He's also an expert in the hematopoietic stem cell biology with a strong interest in RNA modifications and hypoxia pathways in normal and malignant hematopoiesis. Kamil completed his medical degree in Poland. He's Polish by nationality, I think, and did a PhD in biochemistry, University of Oxford, the postdoctoral training in immunology and stem cell biology, also in Oxford. He was a group leader at Oxford and Glasgow and became a professor and a Crook Senior Fellow at the University of Edinburgh from 2013 to 2018. Since 2018, he is a professor of hematology at Barts. And today he's going to talk about targeting RNA modification and hypoxia in cancer stem cells in AML. Kamil?
Thank you, Manel, for a very kind introduction, and thank you all for coming here. It's a really great pleasure to be here in this amazing place. And as you know, as Manel has mentioned, I'm based in the Center for Hemato-Oncology, and having met several people already here this morning, I can clearly see that we really share the same ethos and we've got the same aim of combating blood malignancies, including leukemias. So the central aim of my lab in Barts is to eliminate cancer stem cells in acute myeloid leukemia, and we focus on diverse processes. For example, here today, mostly I will be focusing on targeting RNA modifications and hypoxia signaling and metabolic pathways. Just a very brief introduction. As you all know, hematopoiesis really serves as a paradigm for our understanding of cellular hierarchies in normal tissues and cancers. Stem cells, hematopoietic stem cells, possess unique cell-renewal capacity and multi-lineage differentiation potential and give rise to all blood lineages. In principle, you just need one hematopoietic stem cell to reconstitute the entire blood system. And due to those unique properties, stem cell transplantation offers the best chance of curing multiple diseases, such as blood malignancies, bone marrow failures, genetic diseases, and autoimmune disorders. However, there's a shortage of hematopoietic stem cells for widespread transplantation purposes. And therefore, my lab and many labs in the world are interested in expanding hematopoietic stem cell for transplantation purposes. Acute myeloid leukemia, AML, on the other hand, is an aggressive clonal disease of stem and progenitor cells which acquire multiple mutations to generate leukemic stem cells which fuel leukemia. And the problem is that the currently available chemotherapies really damage normal hematopoiesis, but they fail to eradicate leukemic stem cells which prevail and cause severe relapses with devastating consequences to the patients. So in my lab, like in many other labs, we are trying to tackle this unmet clinical need. We really hope to find ways to eradicate leukemic stem cells without perturbing normal hematopoiesis, or even better, if we manage to eradicate LSCs by with improving normal hematopoiesis. That would be our goal. And obviously, we try to expand HSCs for transplantation purposes. And to address those unmet needs, we are pioneering or planning to pioneer new therapies against novel targets that we have identified as targets in AML to selectively eliminate leukemic stem cells. So in order to achieve this, we have focused on two main programs in my lab. We work on the role of oxygen sensing and metabolic pathways in stem cell biology in leukemia, and this is done in collaboration with Peter Radcliffe and Chris Schofield in Oxford. And another major program we are focusing on is to understand and understand the significance and target RNA modifications in normal and cancer stem cell biology, and this is done in collaboration with Donald O'Carroll at the University of Edinburgh. So first I will talk a little bit about our published work, and then I'll present you some unpublished work focusing on the hypoxia signaling pathway. As we all know, it has become quite well understood that the normal and malignant hematopoiesis happens under hypoxic conditions within the bone marrow. We know that stem cells reside in the most likely the most hypoxic regions within deep inside the bone marrow niches. Stem cells have low metabolic activity, and hypoxia signaling pathways have been implicated in the past in stem cell biology. But how hypoxia regulates stem cell biology and disease such as AML, at that time when we started this endeavor, it was really unknown. So cellular responses to oxygen are mediated by the hypoxia-inducible factor prolyl hydroxylase system. In the presence of oxygen and oxoglutarate, prolyl hydroxylases hydroxylate HIF alpha subunits, resulting in the proteasomal degradation of HIF. However, under hypoxic conditions, when the availability of oxygen is low, for example, in the bone marrow microenvironment, those prolyl hydroxylases, there are 3 of them, by the way, PhD 1, 2, and 3, they are no longer that active, and therefore HIF1 and HIF2 alpha subunits are no longer hydroxylated, but they are stabilized, translocated to the nucleus, where they drive transcription of multiple overlapping and distinct target genes. So we have decided to ask 3 questions here. Do stem cells require intrinsic hypoxia signaling pathways? Does hypoxia and its signaling impact formation and maintenance of leukemic stem cells? And eventually, how can we actually harness this knowledge for therapeutic purposes? So first, I will just describe very briefly this part, because this has already been published quite a while ago. This was done by Amélie and Milica, very talented postdocs in my lab at the moment. Amélie is already a group leader in France. And these 2 talented postdocs said, well, if stem cells reside deep inside the bone marrow microenvironment in perivascular niches, which are very hypoxic, what is the role of hypoxia in stem cell biology? So to tackle this, we decided to delete HIF1 and HIF2 singly and in combination in mice at the same genetic background and ask the question, well, what happens to stem cells? And it was quite obvious in the field that if bone marrow is hypoxic, of course that HIFs will be required for stem cell biology and most likely will have bone marrow failure. But it didn't happen. What we did was to generate HIF1 knockout mice, HIF2 knockout mice, and HIF1 and 2 double knockout mice, specifically in the hematopoietic system, using the VAV-CRE, which deletes in hematopoiesis during embryogenesis. And to our surprise, what we found is that the normal hematopoiesis happened in an unperturbed fashion. We saw unaffected stem cell and progenitor cell numbers, multilineage hematopoiesis, and these mice really aged for 1.5 years, and we didn't see any phenotypes. As you all know here, one of the best ways to enumerate stem cells and test the cell renewal capacity is to do serial transplantation assay, and this is exactly what we did. We transplanted HIF1 mutant, HIF2 mutant, or double knockout stem cells in primary, secondary, and tertiary recipients, and to our surprise, we found that these mice have normal cell renewal capacity and normal multilineage reconstitution potential. So we published these 2 blood papers, which at that point was quite a surprise in the field. But that might not be that bad, because actually we revealed that therapeutic manipulation of HIF1 and HIF2 is unlikely to affect normal multilineage hematopoiesis and stem cell biology if one wants to target HIFs for therapeutic purposes. So the next question we asked is, what's the impact of HIF on leukemogenesis? And again, what we found here was quite surprising and quite the opposite to what was predicted by the literature. So we deleted HIF1, HIF2, and both, and we subjected stem and progenitor cells from these mice to a mouse model of leukemia, where stem and progenitors are transduced with mice1 and HOXA9 oncogenes, which are naturally occurring overexpressed oncogenes in human AML. They are serially replated to form what we call at this stage pre-leukemic cells, which are transformed cells which upon transplantation to mice cause leukemia. So to our major surprise, this is what we found. If you delete HIF1 or HIF2, actually the transplanted mice develop leukemia faster than the wild-type mice. However, if you delete both HIF1 and HIF2, these cells are really very aggressive and they cause leukemia with very short latency. So at this stage, we concluded that HIF1 and HIF2 synergize to suppress leukemic stem cell development and leukemia initiation, and de facto, they are tumor suppressors. Of course, we have corroborated this in multiple different ways. This is all published quite a while ago, so I'm not really going into the details. But for example, here we crossed the HIF2 mutant mice with MLL AF9 translocator mice from Terry rabbits. And again, what we found is that if you delete HIF2, these mice develop really very, very rapid leukemia and very aggressive one as well. So we published this paper in 2015, suggesting that really HIF1 and HIF2 synergize together in AML to suppress the development of leukemia, and we are very glad that friends and colleagues in Lund, including Talia, who is probably here in the audience, they corroborated our data. So really, Talia had a series of papers as well, really showing very similar observation to what we had. So, you know, HIFs have been proposed to be therapeutic targets. We are actually suggesting that they are tumor suppressors. How do we actually harness this knowledge for therapeutic purposes? How can you manipulate these pathways to eradicate leukemic stem cells, or at least compromise them? So, two very talented postdocs in my lab and Chris Cofield's lab. So Hannah is a postdoc in my lab, and Jimmy is a postdoc in Chris Cofield's lab. He's a very talented chemist. They asked the question, well, if you delete HIFs and you get very rapid and aggressive leukemia, what happens if you therapeutically overstabilize HIFs? Can we now eradicate or compromise leukemic stem cells and get rid of leukemia? So this is exactly what they did. And one predominant way to stabilize physiologically HIFs is to inhibit or delete those prolyl hydroxylase enzymes that I've just mentioned. If you delete or inhibit PHDs, then HIF1 and HIF2 stability is constitutively increased. So what Hannah did to start with was to delete genetically different isoforms of PHDs. So we generated PHD1, PHD2, and PHD1 into double knockout mice where the genes are deleted specifically in the hematopoietic system, and we asked, how does this inactivation impact on leukemia generation and development and propagation? So again, we used exactly the same mouse model and various other models that I might not show here. Basically, we took stem and progenitor cells from PHD1 to and double knockout mice and subjected them to this mouse model of leukemia. And what we found is that regardless of whichever PHD you knock out, we can see that the serial replating assay, this assay shown here, a PHD deletion compromises the growth and self-renewal capacity in vitro. And then when you take the equal numbers of those pre-leukemic cells and you transplant them to recipient mice, we can see that the wild-type cells develop very rapid leukemia, but if you delete PHD1, 2, or both, you get very significant, in this case, delay in leukemogenesis. I appreciate that these mice still die from leukemia. However, even several days in this very aggressive model is a big achievement. So then we ask the question, well, as we know, the definition of leukemic stem cells is the ability to propagate leukemia to secondary recipients in this system. So we took the equal numbers of bone marrow cells or equal numbers of c-KIT positive cells which are enriched for leukemic stem cells, and we transplanted them to secondary recipients. And what we can see already here is that if you delete, in this case, we've done it for PHD1 knockout, we can see that if you lose PHD1, you really ablate the capacity of these leukemic cells to cause aggressive leukemia. So PHD1 is essential for leukemic stem cell self-renewal and leukemia maintenance. So then we asked another question. What happens if you ablate PHD2 or inactivate PHD2 in established leukemia? So we took advantage of a mouse model that was published by Peter Radcliffe. We are part of this study a couple of years ago, several years ago. In this case, these mice express short happen against PHD2, which is tamoxifen inducible. So we took stem and progenitor cells from these mice that have never seen, sorry, it's doxycycline inducible. They have never seen doxycycline at this stage. We transformed them with mice 1 HOXA9, injecting them to recipient mice and allowed them to develop leukemia. We can see here at day 60, both control and PhD knockdown that have not received the knockdown yet develop equal leukemia. And at this stage, we say, yes, these mice develop leukemia at diagnosis, and then we introduce the doxycycline to knock down specifically PHD2. And what we can see already is that the control cells progress to develop leukemia, but the cells with the knockdown for PHD2 slow down, and they eventually cause leukemia with a very delayed latency. And here in this model, delay in 50 days in this very aggressive model is to our knowledge quite a significant achievement. So how do we inhibit PHDs pharmacologically? How can we harness this for patient benefit in the future? So this slide here shows the PHD2 mediated catalysis. We can see that HIF alpha subunits have 2 proline residues, and in the presence of oxoglutarate and oxygen and ferrous iron, proline hydroxylases hydroxylate those prolines, and that results in HIF degradation. So how can we inhibit it? Well, there are at least 5 different clinically available compounds which are available for renal anemia. So we thought, yeah, let's use these compounds and see what happens. So what we know about daprodustat, for example, is that it's very similar structurally to 2-oxoglutarate, and it really displaces oxoglutarate from the active side of PHD, thus inhibiting the enzyme. And here's the structural basis for this. So here we can see the overall, we can see the catalytic domain, catalytic pocket of proline hydroxylase, and we can see the 2-oxoglutarate pocket here. Here is the 2-oxoglutarate, which is a co-substrate of this enzyme. It chelates the ferrous iron, and it sits very comfortably in this pocket. But if you add the daprodustat displaces and takes the place of 2-oxoglutarate, thus inhibiting the enzyme. And that applies to the inhibition of PHD1 and 3 as well. However, when we looked at the daprodustat in more details, we realized that it does very strongly inhibit, here is the IC50 data, very strongly inhibit PHDs, but it also inhibits other 2-oxoglutarate dependent oxygenases, including JMJD6. And that's not what we really want to do in leukemia, because we had a paper a couple of years ago now showing that JMJD6 promotes the self-renewal and regenerative capacity of stem cells. So that's the last thing you want to do, is to inhibit JMJD6 in leukemia. So we came up with a brand new compound, which we just filed the patent for. And that was generated by Jimmy and Chris Coffey's lab. And we are optimizing this compound to become a clinical grade inhibitor. But it was designed in a completely different way than the daprodustat was. So we call it IOX5, inhibitor of oxygenases number 5. They generated 1, 2, and 3. Now we've got a 5. And the way that it binds is that it just binds at the entrance to the catalytic part of the enzyme. And as you can see, it uses completely different residues compared to the daprodustat, giving it specificity. Those 2 key tyrosine and arginines are no longer engaged in the binding. And this compound was designed in such a way to mimic the way that HIF enters this domain to be hydroxylated. So it really blocks the binding between HIF and PHD. And we could see here that this compound is far much more selective. It specifically inhibits PHDs, but has no activity on other oxoglutarate-dependent oxygenases that we have tested. So what we know about these 2 compounds, the daprodustat and IOX5, is that they very strongly stabilize HIF, as expected. And we can see that they have very strong anti-proliferative and pro-apoptotic capacity in mouse and human AML cells. Here we can see a panel of different human AML cell lines with diverse genetic mutations, and we can see that both the protostat and IOX5 block the proliferation of leukemic cells and induce massive apoptosis. So then we wanted to see whether this compound is actually active in vivo, and we really have promising data. This is work in progress. We can see that we basically engrafted THP1 cells, which is a cell line for MLLAF9 translocation. We engrafted that in mice, and we treated these mice upon leukemia development with the protostat and IOX5. And we can see that these two compounds are not toxic to these mice. The mice are quite well tolerated, but it does decrease the leukemic abundance and engraftment in these mice. So then we ask the question, well, is this an on-target effect? The prediction is that if you inhibit PHD1 or 2, you have stabilization of HIFs, and that would result in, that would compromise AML. So what happens if you delete HIF1 and HIF2? Is this mediated by HIFs? And indeed it is. So here I'm showing the data where you have HIF1 control and HIF1 double knockout cells, and control cells that are incubated with IOX5. So the control cells that are incubated with IOX5 slow down the proliferation and eventually die. But if you delete HIF1 and HIF2, you completely rescue this effect. So we think that indeed this is an on-target effect where inhibition of PHD results in activation of HIF and eventually compromises AML. So then we wanted to understand a little bit about molecular mechanisms through which all this happens. So we took leukemic cells with MLLAF9 translocation, treated them with IOX, and did RNA sequencing. And we could see is that a massive upregulation of a huge number of HIF targets here. So basically the genes that are upregulated by IOX are the HIF targets that are positively regulated by HIF. And the genes that are downregulated by IOX are the ones that are normally downregulated by HIF. We could see here is that IOX5 really reprograms the metabolic properties of leukemic cells, and this is all rescued by HIF deletion. So we can see other transcriptional programs that are permissive for blocking leukemogenesis, and one of those key pathways that we identified was obviously significant upregulation of apoptosis pathway, which we have proven at the biological level. And one of the key pro-apoptotic genes that we know is a HIF target is BNIP3. So we looked a little bit more at BNIP3. We can see that IOX5 is upregulated, this pro-apoptotic gene in different human AML cells. So this is still a working hypothesis, but what we think is happening is that if you inhibit PHDs, then HIF is upregulates its target gene BNIP, which then triggers apoptosis through BAX and BAC system, which depolarize the outer membrane of mitochondria. What we do know in AML as well is that BCL2 is always hyperactive, and it kind of inhibits the apoptosis of leukemic cells, again by inhibiting BAX and BAC mediated apoptosis. So we ask the question, what happens if we inhibit PHDs and also co-inhibit BCL2, which we know is a target in AML, and venetoclax has been added to multiple different treatment regimens. And what we found was really quite amazing. We found that if you treat leukemic cells with IOX5 and venetoclax, that really results in a massive apoptosis of diverse AML cells, mouse and human. Here are some preliminary data in patient samples. Here are the CD34 enriched AML cells, and from patients, and we can see that again, IOX5 and venetoclax, if not synergistically, at least in additive fashion, they kill the immature leukemic cells. And I must add that these samples are from P53 mutant AML patients which are very difficult to kill by any known means. So are PhDs therapeutic targets? If they are, that would be amazing, but what is the impact of PhD inhibition or inactivation on normal hematopoiesis? So this is exactly what we did. We inhibited PhD, knocked out PhD in normal hematopoiesis, and we really saw no major defects. The hematopoiesis happens quite normally in these mice. We transplanted bone marrow from these mice, and again, we could see that the stem cells lacking PhDs really reconstitute hematopoiesis in a normal way. So we think that PhD inhibition stabilizes HIV to compromise AML through BNIP, but has little effect on normal hematopoiesis. So which AML subsets is sensitive to PhD inhibition? This is really not known. We are looking at diverse AML subsets, trying to identify those where we can really eradicate leukemic stem cells. What are the mechanisms via which HIVs compromise AML? BNIP is one of them, but probably there are many other ones and we need to identify. And what are the synthetic lethal interactions between PhD inactivation and inhibition of other targets? This is that sort of thing that we are planning to develop using CRISPR-screen approaches. So as you know, PhDs belong to a group of enzymes called alpha-ketoglutarate-dependent oxygenases that hydroxylate HIV, but there are many other similar enzymes that hydroxylate different substrates like transcription factors, RNA, splicing factors, ribosomal proteins. So we are really very interested in identifying those oxyglutariate-dependent oxygenases that have impact on stem cell biology. Are they important? Are they druggable? Can we kill leukemic stem cells by inhibiting all these enzymes? And we are really working on them. And some of those enzymes are erasers of RNA modifications. So this is how we entered the field of RNA modifications. Together with Donald O'Carroll in Edinburgh, we investigated the significance of different RNA modifications in AML. So as we know, gene transcription is really critical for stem cell maintenance, and the dysregulation of transcription might result in leukemogenesis. But what has been ignored for quite a while is that many mRNAs undergo chemical RNA modification. So the significance of many RNA modifications in normal and malignant hematopoiesis remains unknown. And this obviously gives us an opportunity for brand new discovery with a potential therapeutic development. So this is RNA as we know it. It contains stop codon, starts with a cap, finishes with body A tail. However, what has been ignored for a very long time is that every single residue of RNA could be modified by different chemical modifications. And what is the functional significance of those RNA modifications in AML really remains unexplored. So we have focused on M6A modification. M6A is the most abundant internal RNA modification installed by writers and removed by erasers. And the function of M6A is executed by readers, which recognize M6A modified transcripts to promote RNA decay, translation, or splicing. We have focused on 3 such readers: YTHDF2, which promotes RNA decay, and YTHDF1 and 3, which promote RNA translation. However, more recent data suggested that these readers are actually redundant and they promote only RNA decay. So what we found is that YTHDF1, 2, and 3 are overexpressed in diverse AML subsets, and indeed they are overexpressed in leukemic stem cells compared to their non-malignant counterparts. So together with Donald O'Carroll and multiple people in my and his lab, we asked the question, what is the role of YTHDFs in stem cell biology and leukemia? So to start with, we focused on YTHDF2 because it was consistently overexpressed in AML and its expression correlated with leukemic stem cell activity. We found that YTHDF2 at the protein level is very highly expressed in AML. So we asked the question, what is the significance of YTHDF2 in AML? And we took exactly the same approach. We generated YTHDF2 knockout mice and transformed stem and progenitor cells in a mouse model of leukemia. And what we found is that really regardless of the model of leukemia we used, YTHDF deficient cells failed to undergo efficient transformation. When we transplanted those pre-leukemic cells to mice, we found that YTHDF2 mutant leukemic cells really had generated leukemia with very, very extended latency. 50 days or more in this mouse model is very significant delay in leukemia. And then we did the limiting dilution assay. I don't have time to discuss it, but basically we arrived at the conclusion that the frequency of leukemic stem cells was decreased upon YTHDF deletion. And then we corroborated this in a completely different model system where we didn't use the retroviral model system, but we took the YTHDF2 knockout mice and crossed them to the inducible MLL-AF9 mouse model. And we can see here, without going to technical details, if you delete YTHDF2 in the MLL-inducible mouse model, this almost completely rescues the mice from long-term leukemogenesis. Then we ask the question, what happens if you delete acutely YTHDF2 in established leukemia? So we generated leukemia through primary transplantation and then transplanted leukemic stem cells from primary recipients to secondary recipients. And upon leukemia diagnosis, we inducibly deleted YTHDF2 using MX-CRE system. And we can see here that this flat line, red line, deletion of YTHDF2 from established AML almost completely rescued the mice from the disease. So we ask the question, is this significant? Is this significant in human system? So we knocked down the expression of YTHDF2 in diverse cell lines, and what we really found, as shown here, they slow down the proliferation, and the knockdown of YTHDF2 results in apoptosis of leukemic cells. And this was exactly the case in human samples. We can see here that if you knock down YTHDF2 from AML patient samples, it almost completely obliterates the ability to generate leukemia. So then we wanted to understand the mechanism through which all this happens. We did RNA sequencing and M6A sequencing where we found those transcripts that the M6A modified. And without going into major details, what we found here is that genes that are significantly upregulated upon loss of YTHDF2 have M6A occupancy. So if you delete YTHDF2, you get overstabilization of those transcripts that are modified by M6A. And we also did the SLAM-seq, which is the global RNA stability assay, and this is exactly what it's showing, that those transcripts that are M6A modified have increased lifetime. Half-life. So we concluded that the YTHDF inhibition results in stabilization of those transcripts that are M6A modified. And we interrogated those transcripts using multiple different ways. I don't have time to go to the details, but what we found is that those transcripts that are upregulated upon YTHDF2 deletion are also upregulated in AML samples that have lost the capacity to generate leukemia upon transplantation. So in those weak leukemic cells from human patients, we can see the same signature as upon YTHDF2 deletion. And one of those transcripts was TNF receptor 2. So we looked into details about TNF receptor 2, and what we found is that it was highly M6A modified in mouse and human cells. We found that YTHDF2 indeed binds the TNF receptor transcript and degrades it. TNF receptor is obviously a cell surface molecule, so we can see here by flow cytometry that if you delete YTHDF2, TNF receptor 2 is highly upregulated. Then we asked the question, are these cells more sensitive to TNF? So we took wild-type and knockout cells and we treated them with TNF, and what we found is that if you add TNF to the knockout cells, they are really sensitive to TNF treatment and they undergo massive apoptosis. So we think that it might well be possible that inhibition of YTHDF2 with TNF could be a potential therapeutic strategy in AML. So if YTHDF YTHDF2 was a therapeutic target, well, what is the impact on normal hematopoiesis? So we decided to delete YTHDF2. I will skip this. We decided to delete YTHDF2 inducibly using MX-CRE and follow these mice for 1 year. And what we really found is that apart from slight myeloid bias, these mice generated or sustained normal hematopoiesis in a long term. And we can see here in young mice, now and going back to the VAV-CRE knockout system, we can see that they have decreased white blood cells and a little bit of red blood cells, but deletion of YTHDF2 doesn't really derail normal hematopoiesis. But what we do see is that we see stem cell expansion. And when we age these mice for more than 1 year, actually they do sustain normal multilineage hematopoiesis. We don't even observe those defects that we saw in young mice, but we can still see expansion of stem cells. So then we transplanted those stem cells from young mice, and we found that overall they have normal reconstitution capacity with a myeloid bias, but they do lose T cell capacity. But overall, the stem cells lacking YTHDF2 have enhanced reconstitution potential in stem and progenitor cells and in mature cells. However, if you transplant them to secondary recipients, this is where we see serious phenotypes. We see that YTHDF2 mutant stem cells fail to undergo cell renewal upon serial transplantation. So we think that YTHDF2 protects stem cells from exhaustion. And looking at the mechanisms through which that might happen, we did RNA sequencing here, and we found only 81 transcripts being upregulated. But the majority of these transcripts actually represent pro-inflammatory pathways, like interferon pathways, TNF pathways, pro-inflammatory response. And we obviously know that inflammation inhibits cell renewal and causes exhaustion. So to look deeper into the mechanisms, we performed M6A sequencing in stem and progenitor cells. And what we found is that upregulation of those pro-inflammatory genes belonging to TNF pathway, interferon pathway, and STAT1 correlates with M6A modification. So those transcripts that are M6A modified, they are upregulated upon YTHDF2 deletion. And here we can see the evidence of that. We can see there are several different transcripts which are pro-inflammatory transcripts that are highly methylated. So we think that YTHDF2 inhibits pro-inflammatory signals to promote cell renewal and prevent exhaustion. So is YTHDF2 induced by inflammation itself? So we took advantage of the reporter mouse in which GFP is fused to YTHDF2, and we treated these mice with poly-I, poly-C. And what we found indeed is that YTHDF2 was very highly upregulated by inflammation. So we think what's happening is that in pro-inflammatory stimuli, YTHDF2, what pro-inflammatory stimuli do is to activate pro-inflammatory pathways, which triggers the emergency hematopoiesis. However, YTHDF2 is one of the negative feedback loops that dampens those pro-inflammatory pathways. And if you remove YTHDF2 in the longer term, that triggers the exuberant pro-inflammatory pathways, and instead of causing healthy emergency hematopoiesis, that enhances stem cell aging and loss of function. I'm running out of time, actually, so I probably have to skip a few slides. But actually, what I can do, really, I mean, do I have a few minutes, or shall I? Yeah. What we think is that YTHDF2 is a therapeutic target in AML, and inhibition of YTHDF2 might compromise leukemic stem cells, but in a shorter run, it will expand normal stem cells, and maybe in a longer run, it might have detrimental consequences to stem cell functions. So how can we target YTHDF2 therapeutically? What is the M6A binding domain of YTHDF2 required? So here we did a genetic experiment where we asked the question, is targeting the YTHDF2 sufficient, the YTHDF2 binding pocket sufficient to inhibit its function? So here is the structure of the M6A binding aromatic pocket within the YTHDF2. We can see those key tryptophans here, which very tightly bind the M6A modified transcripts. So to address the question whether this aromatic pocket is required for YTHDF's function, we decided to generate a point mutation where this green tryptophan was replaced with alanine. And in this case, this mouse expresses YTHDF2 in such a way that it does not bind the M6A modified transcript anymore. So he asked the question, what is the impact of genetic disruption of the YTHDF M6A binding on normal hematopoiesis and malignant and AML? And this is exactly what we found. So we generated mice, which are control mice. Those completely lacking YTHDF2 in the hematopoietic system, and those WA mice, we call them WA because it's a tryptophan to alanine substitution. And what we can see here in brief is that those WA mice really phenocopy the mutant. We can see LSK cells expansion, stem cell expansion, and those multipotent progenitors are expanded if you delete YTHDF2 or just ablate this binding to M6A. What happens in the leukemia system? So we took those WA mice and we crossed them to inducible MLL mouse model, and we did a simple proliferation assays. There are many in vivo assays going on at the moment, but I don't have the data to show you. But what we can see here is that if you ablate the ability of YTHDF2 to recognize M6A, that results in the inability of these cells to proliferate and generate colonies upon serial replating. So I will conclude my talk here, I think, because I've just run out of time. But what I'd like to say is that we think that YTHDF2 is a good therapeutic target in AML. Targeting YTHDF2 domain of M6A binding domain is sufficient to inactivate YTHDF functions. So the question is now, can we target this therapeutically? We are at the moment launching the drug discovery program to identify small molecules against YTHDF2 and several other readers that we are focusing on. And hopefully I'll be able to report a bit more data in the near future. So I'd like to thank my hardworking group in London and my key collaborators on this program, Peter Radcliffe and Chris Schofield, who really helped me a lot to develop the hypoxia part, and my friend Donald O'Carroll in Edinburgh, who really introduced me to the M6A and mRNA modification system. And obviously, I'd like to thank my funders for the generous support of this work. And thank you so much for your attention.
Thank you very much. Thank you very much, Kamil, for this wonderful lecture. There's a joke here. So, inhibition of PhDs against AML, but the stimulation of PhDs for AML research. No? So, one question, there are several considerations. One is, how redundant are the reader of M6A? Because you said this at the beginning, no? So, do you think that this is relevant, or you obtain similar results with the other M6A RNA binding proteins?
Yeah, I skipped that part of my talk, a few slides, because I just didn't want to run out of time. But we deleted YTHDF1 and YTHDF3, and we see a very similar effect. They are not as strong as with YTHDF2, but there's a therapeutic possibility or opportunity here, because if you delete YTHDF1 or 3, or both at the same time, you still manage to kill leukemic cells, but there's absolutely no consequence to normal hematopoiesis. So maybe there's even a better therapeutic window for targeting other readers. But YTHDF2 is obviously one of the first ones we tried. Potentially the other ones might be even better.
And for the PhD inhibitors there, so it does matter the genetic configuration of the AML, or it does not?
Well, we have focused initially on P53 mutant because we had a lot of those samples, and they are obviously one of the most difficult to eradicate. But then we looked at nucleophosmin mutants in combination with FLT3, which is the intermediate prognosis leukemia. And here we can also see a very significant effect of PhD inhibition. But the beauty of the FLT3 ITD mutation is that you can also use gilteritinib, which is the inhibitor of FLT3. And we really saw a very nice synergistic effect between IOX5 and gilteritinib. So in this case, taking advantage of the genetic alternations like FLT3, we can generate possible new therapies that are combinatorial and synergistic. But we are looking into greater details at different other genetic subtypes of AML.
Thank you. Questions?
Thank you. Thank you, Kamil. Very, very nice work. Congratulations for all this work. I have a question. Maybe I missed it. I have a specific question and then a more general question. But do you, when you do the transplantation of the PhD deletion in the normal setting, have you done serial transplantation in that?
Yes, I haven't shown here for the sake of time, but we have gone into tertiary transplant, primary, secondary, and tertiary.
And no effect.
No effect, no.
Long term.
So, I mean, again, there's a lot of work, and I'm summarizing it in one slide, but we deleted YTHDF1, PhD1, PhD2, and both. We did serial transplantation, and there was absolutely no phenotype. But there's this one thing that I missed here, that I didn't show here, is that if you delete both PhDs, you actually have expansion of stem cells, and this is non-malignant expansion. They do generate multilineage hematopoiesis. So it could well be that, you know, by dual inhibition of PhD1 and 2, you might not only slow down leukemic development, but actually boost normal hematopoiesis. We are working on this. I didn't show these data here, okay. But we did extensive.
And my other question, which is more philosophical, is that a lot of, I mean, we all work with similar models, and we're targeting proliferating cells. And when you slow down leukemia, or survival, let's put it this way, how do these cells, do you think, or if you have tested, how would these cells react on chemotherapy, for example, on therapy resistant? Are we making more resistant cells, or are we curing leukemia?
It's a very good point. I didn't elaborate on this. When we show slowdown of proliferation, is really a growth assay, but the curve becomes flat because of the apoptosis, not because of the slow proliferation. We did extensive study in YTHDF mutant and in PhD inhibitors. We didn't see any cell cycle arrest, or we didn't force these cells to quiescent. And yes, we used, and well, these are relatively preliminary data with patient samples, but we are using IOX5 inhibitor for PhD with diverse therapies like venetoclax or different combination of the known chemotherapies. And so far we see either a synergistic or additive effect. But we are now launching, the proper PDX models in this system in vivo, so this will be very telling. So these are early studies, but I completely take your point that we don't want to induce quiescence of leukemic stem cells.
Carmen here. So first of all, thanks a lot. That was very enlightening. I want to hook up on the very last few slides where you actually explain this upregulation of inflammatory pathways. So we very often see this whenever we have something which is slowing down leukemia. So are we sure this is actually the cause of the effect, or maybe a consequence because we are differentiating hematopoietic stem cells into immune cells or more mature immune cells?
So you're asking whether the upregulation of the pro-inflammatory stimuli in
cause
HSC...
or consequence?
We don't know. That's the answer. Reviewers asked us to cross these mice to multiple different mutants to show the causality, and we are not able to do it. This paper was published actually in the middle of pandemic, but we are looking into this probably more pharmacologically. We plan to inhibit those pro-inflammatory pathways using small molecules that are available to us and see if we can rescue the phenotype. If that was the case, then I would say it's causal. At the moment, you've got a point. It's very difficult to say because we've got many of these transcripts being stabilized, 81. It's very difficult to decide which one to target to show the causality. But I take your point.
I also wonder, I'm not sure if anyone has done this, to go to really inflammatory diseases and just to repeat the leukemia model in an inflammatory context to see how this actually would look like.
Yeah, that's another point. I mean, it has been suggested to me before that, MDS could be a pro-inflammatory disease where pro-inflammatory stimuli cause MDS. So therefore, I got questions from reviewers quite rightly. Do you actually elicit the MDS-like phenotype in these mice or any other models that we've got? And the answer to this was no. We really took those questions seriously. We aged these mice. We looked at the histology. We in different stages we did sequencing. We didn't see any dysplastic phenotypes. So at least in this context, if we were to inhibit YTHDFs, that wouldn't probably result in any MDS-like phenotype in the longer run.
Oh, thanks.
Nice talk, Kamil. Do you think that this therapeutic approach of targeting RNA modifications and hypoxia signaling could be applied to the other big group of acute leukemias, the acute lymphoblastic leukemias?
That's a very good question, and we are indeed looking at this. I don't know about ALL, but I think there are a few papers actually in circulation. They are probably not published yet, indicating that readers will be important. Certainly, I haven't shown these data, but at least in mouse models and in cell lines, human cell lines, we have depleted YTHDF2, and we showed really very spectacular apoptosis of multiple myeloma cells. And we crossed the YTHDF2 mutant mice to CLL model of CLL, and we could see that it has also a very significant effect. So I think that whatever I have shown in AML will have benefit in other leukemias, but we have focused on AML to start with. We obviously very keen to look at ALL and other diseases. But another thing to say, I'm not sure yet about the hypoxia part because we haven't done any of these studies, but we can see that a proliferation of literature about YTHDFs being targeted in multiple different cancers, in breast cancer, in pancreatic cancer, in prostate, esophageal. So I'm quite confident that by inhibiting or making a small molecule inhibitor for for YTHDFs, we will provide possible treatment strategies for other solid tumors.
Thank you. Thank you for the talk. So I have just two questions. One is about HIF, if there are mutations prevalent in AML that can be found. And the second one is if you have tested the IOX5 inhibitors against AML cells with mutations in IDH that has altered the isoacetylglutarate metabolism.
Well, the quick answer to both questions is no, we haven't done that. So HIF is not mutated in AML, and we looked quite seriously at the publicly available data, and it's not mutated in any way. And no, we haven't done the IDH study. That's something on our radar that we hope to do, because obviously IDH produces 2-hydroxyglutarate, which might affect the outcome. But that's a very good point. We should look into that in the future.
We should close here. Thank you, speaker, again.
Okay.